Hello friends, this video on cell cycle and cell division part 9 is brought to you by examfear.com. No more fear from exam. As I said, cytokinesis is the final step where the cytoplasm will get divided into two daughter cells. So here the cytoplasm of parent cell divides into daughter cells. So in the telophase itself, it indicates the beginning of cytokinesis with this furrow. So what is this furrow? Now, the process of cytokinesis happens differently in animal cells and in plant cells. So in case of animal cells, cytokinesis happens with the formation of a furrow. What is a furrow? You can see here from the periphery of the cell, a structure like this is formed. Now this will gradually deepen inside and it will break the cell into two halves. Now what is this structure actually? This is nothing but a ring of actin, which is a protein. So a fiber ring made of protein called actin is formed around the center of the cell. Now this ring is somewhat like this. Let us suppose a ring like this. Now just imagine a bangle or something like that in the middle of a balloon. Now if you keep reducing the size of the bangle, what will happen? This width will keep on reducing. Let us suppose if initially it was like this, you keep a bangle here and tighten the bangle. So what will happen? A furrow will develop like this. You tighten the bangle even further, so it will be like this. If you tighten it even more, it will break into two parts. So exactly that is what is happening here. So that fiber ring made of protein called actin around the center of the cell, it keeps on contracting. When it contracts, the cell pinches it into two daughter cells. Now when it divides into two daughter cells, this half will become one daughter cell, this half will become one daughter cell. So both of them have got separate nucleus with a separate nuclear membrane. Both of them have one set of chromosome each. Right? So the process of cytokinesis happens successfully in case of animal cells. So you get two daughter cells like this, which are exactly identical to each other. Why are they exactly identical? Because both of them have got the same daughter chromosomes. I mean, they have got just one sister chromatid is with this and the other sister chromatid with this. So basically they have the same chromosomes. So now you understand how the process of mitosis is based on just two words: replicate during the S phase and divide during the anaphase. So that is mitosis. Now, as I said, in case of plant cells, cytokinesis happens in a little different way. Why? Because when you talk about plant cell, they have an additional cell wall outside their cell membrane, and that cell wall is made up of cellulose. Now, as I said here, in this case, this actin ring actually helps to create the furrow. But in plant cell, that is not possible, because in plant cell, the presence of the cell wall makes it very stiff. So it is not at all flexible that it can be contracted with the help of the actin filament, actin fibers. So they cannot be constricted by actin fibers, and therefore in plant cells, what happens? This is the plant cell. So the green color structure, this is the cell. I have not drawn the internal structure. I am just explaining the concept. The yellow colored boundary which you see, that is nothing but the cell wall. So what happens is the vesicles inside the cell form a membrane partition like this. A cell plate is formed like this. And then this plate gets divided from between, and it forms two cells. So from where does the cell plate formation starts? It starts to form from the center, and then it gradually spreads on both the ends, and then finally it breaks it into two halves. So here there is no role of the furrow or no role of actin fibers, because the cell wall is very stiff. An additional cell plate will be formed starting from the center, and then it will finally split the parent cell into two daughter cells. So that is how cytokinesis will take place in plant cells. So now that we have understood the different steps of mitosis, let us quickly look at the overall process of mitosis. So now I'll try to make use of some animation so that I can explain you it in a better way. So this is from where we started the process of mitosis. What was there? We had the chromatin, which were not condensed into chromosomes, just the chromatin fibers, right, which have already been duplicated during the S phase. Again, we had the centrioles, which were duplicated during the S phase. So right now we are in the prophase. So in prophase, we have the duplicated chromatin threads, we have the duplicated centrioles. So what happens during prophase? The chromatin condenses to form chromosomes. So chromosomes, they are distinct structures shaped like X. At the center, they have centromere and kinetochores, which are nothing but proteins on the outer part of centromere. Also, what happens during the prophase? The centrioles start to move towards the opposite poles. So centrioles have moved towards the two different opposite poles, and spindle fibers, or these microfilaments, microtubules, will start to form from both the centrioles. Now, some of these microtubules will be kinetochore microtubules, those which are connected to the kinetochore. Some of them, for example, this one, if you look at this one, it is not connected to any kinetochore. So this will be known as a non-kinetochore microtubule. Now, these microtubules, the counter forces between the microtubules will align all the chromosomes at the equator, and this is known as the metaphase plate because it occurs in metaphase. Now, when all of them are aligned at the equator, they are aligned in such a way that one sister chromatid is connected through the microtubule of one pole, whereas the other sister chromatid is connected through the microtubule to the opposite pole. Now, the kinetochore microtubule, that means these microtubules, will pull the sister chromatids towards each other, and they will try to push them towards the two opposite poles. You saw that? We'll see it once again. So, both these kinetochore microtubules will pull the sister chromatids towards their own poles. So as a result, the sister chromatid, one sister chromatid will move towards this pole, the other sister chromatid will move towards this pole. So the sister chromatids will get separated, and then what will happen? The cell will also elongate due to the non-kinetochore microtubule. Now, once the sister chromatid comes on the two opposite poles, nuclear membrane will develop and a furrow will develop, which will cause cytokinesis. Right now, we are talking about animal cells, so a furrow will develop like the way it has been shown here. So, once again, just look at this tape. Chromatids getting separated, so now they become daughter chromosomes, and then from the periphery this furrow develops, which finally divides the cell into two halves. So now you have two daughter cells which are exactly identical to each other. So this is the overall process of mitosis. Now please understand the process of mitosis very clearly, because if you understand mitosis, meiosis will become quite easy, because you will know the basic concepts that how these things happen. Okay, so with this we'll end our discussion on mitosis. But before we start with the next process, that is meiosis, let us quickly discuss the significance of mitosis. It is restricted mostly to diploid cells. So when I say restricted to diploid cells, that means it takes place in all the diploid cells present in our body. So because of the mitotic division, the diploid cells of our body can increase in number. For example, our growth, or when you look at the growth of a human being from a small fetus, that all happens by mitotic division of the diploid cells. Cell growth in case of both plants as well as animals. Cell repair. Whenever there is some injury or damage, or whenever a cell dies off, so that the replacement is always provided by mitotic cell division. Regeneration of body parts. For example, there are some organisms which can regenerate their lost body part. For example, starfish, they can regenerate their lost arms by the process of mitosis. So if it loses this arm, it can produce it again. That happens by mitotic cell division. It helps in asexual reproduction. So where you do not need separate sexes to reproduce, like you do not need a separate male sex cell and a female sex cell to reproduce. So for example, in case of Hydra, how do they reproduce? They reproduce by a process called budding. What happens in budding? A small outgrowth develops, which is known as bud, on the surface of organism, and then gradually it grows and develops. When it becomes mature, it gets separated from the parent organism. So that is how they reproduce. Now if you see, from where does this outgrowth come? So from where does this extra cell for the outgrowth comes? That comes by mitotic cell division. So mitosis also helps in asexual reproduction. That is why I had told you before that mitosis is more related to asexual reproduction, whereas meiosis is more related to sexual reproduction. So with this, we will end our discussion on mitosis. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.